Protein synthesis has two parts. The first part is transcription. Transcription has 4 steps, the first of which is initiation. In initiation, RNA polymerase binds to the promoter region with the help of transcription factors, or proteins, and the TATA box. The polymerase begins to unwind the DNA, and the second step, elongation, begins. In elongation, the DNA is unwound, and RNA nucleotides are paired to the DNA gene sequence. RNA is transcribed in the 5' to 3' direction. The RNA will peel off and DNA will reform as the polymerase continues. Once the polymerase reaches a sequence that triggers its dislocation, the third step, termination, is started. The polymerase passes the termination point before the RNA is actually released. Prokaryotes have 2 methods for termination. The final step is mRNA processing, which only occurs in eukaryotes. In mRNA processing, a 5' cap is added, as well as a poly A tail. The spliceosomes, made of SNRPs, remove introns. The exons may be shuffled, which can lead to the evolution of new proteins. Once mRNA processing is complete, the mRNA, called mature mRNA, is ready to leave the nucleus and enter the cytoplasm, leading to the second step of protein synthesis, translation. Translation has 3 steps. The first step is initiation. Prior to initiation, tRNA receives its amino acid from an enzyme called aminoacetyl tRNA synthetase. During initiation, the small subunit of the ribosome attaches to the mRNA at the start codon, AUG. tRNA, with the anticodon, UAC, will attach to the P site, and the large subunit will attach to the small subunit. After this elongation occurs, the mRNA moves down one codon and the tRNA moves from the P site to the E site. The tRNA at the A site then moves to the P site, and a new tRNA fills the A site. This process continues, adding more amino acids, forming a peptide bond through dehydration synthesis. GTP is required in this process. When the mRNA reaches its stop codon, UAG, UAA, or UGA, a release factor attaches at the A site. This causes the release of tRNA, the polypeptide, and the ribosome pulls apart.